Okay, so I think, come, Kubi, we took an early break. So.
Oh, I didn't know that.
But you are really early for your lecture, so.
Okay.
I'm going to 15 minutes, okay?
Okay.
So, I think what I'm going to do is I'm going to talk about one important thing, and then Prem brought up a small point that I want to address so that you know a little bit about how these things are written. So, and not really do a whole lot about the polysaccharides that I have for this particular lecture. Okay, so one point about disaccharides obviously is that the monosaccharides are connected through a bond. Okay, so the OH, for example, here is connected to a carbon, and this is called the interglycosidic bond. Yeah, and that's important for polymers. The interglycosidic bond is the one that connects monosaccharides. The numbering 1 and 2 essentially refer to carbon number 1 is connected to carbon number 2 for the second monomer. That's 1. The alpha refers to that particular bond which is identified through orientation with that CH2OH. Yeah? Right? It's essentially the same thing that we discussed earlier. So this bond is away from that particular OH, and therefore this is alpha. Okay? So that's how alpha 1,2 bond, and likewise you can have for all the rest of the chain. Then Prem asked about, let's see, where do I have that? I guess in the first slide. Yeah, Prem asked about this particular letter. So notice that this is, so this stands for D-galactosamine. This P is the pyranose form, right? Is a pyranose form that we discussed, the 6-membered ring, the cyclic 6-membered ring. If you had an F, that would have been the furanose form. That would be F. 2 is, at position 2, you have an acetyl group. That's what this means. That means this is normally GLN-N, this N is always at position 2. So we say this is N-acetyl. And then alpha 1,4, okay? 1 to 4. That means position 1 is now connected with position 4. Okay. Alpha D, which is what we have identified, P pyranose, and rest of the chains. Okay. So in essence, that is how the polymers are built up, or that is how the oligomers are built up. One last point about the structure that I think I want to point out is the following. So here, for example, this is lactose. Lactose is a combination of beta-D-galactose and D-glucose. The interglycosidic bond is beta 1,4. So this is 1, position 1 connected with position 4. This is orientation similar to the similar to 6 CH2OH, and therefore this is beta. So this is beta 1,4 interglycosidic bond. And this is the overall geometry, the three-dimensional geometry. Obviously, we talked a little bit about lactose intolerance arises from an enzyme deficiency, lactase, that hydrolyzes this bond and subsequent problems thereof. So I want to ask you this question. So if you have lactose, for example, D-galactose, beta-1,4, D-glucose, is it the same as D-glucose, beta-1,4, D-galactose? I think you all know the answer, but I want you to just think a little bit about what this is. So what are the difference? The difference is that galactose and glucose has been swapped. The two have been swapped. Normally, and I think I forgot to mention, normally what do? We have the reducing, we didn't talk about reducing terminus. So hopefully Kubi is going to talk about it a little bit later. But in essence, the aldehyde, this particular group is called the reducing terminus, right? So this particular end is called the reducing terminus. This end is the non-reducing terminus, okay? And there is a reason for that. So Kubi will begin that. I had it on the characterization slide, but I'm not going to start that now. So the way that this is written is that this usually, the residue that is written on the rightmost part is usually the reducing end of the molecule. So glucose actually has a reducing end. So this is actually an aldehyde, right? This is the non-reducing end. So in this case, galactose is the reducing end. And here glucose is the non-reducing end. So that's the difference. You are swapping. Are they the same? I think you already know the answer. They are not the same. Okay? They are not the same. They are not only just not the same because there is a swap, but if you look at the structure of the two molecules are completely different. Structure of the two molecules are completely different. So by swapping, you're actually changing. You may not be changing the residue type, but you are actually changing the three-dimensional geometry dramatically. It's completely different. So much so, and I did mention that, that even if you have D-glucose and L-glucose, one of them is a biologically important molecule, the other one remains untouched. Not only that, you have glucose and you have galactose, okay? This is position 4. Notice, at position 6, both are the same. At position 2, both are the same. At position 3, both are the same. This is galactose and this is glucose. At position 4, one of them is an axial, the other one is equatorial. And the body can recognize that. Even single change can be recognized. Okay? That's pretty phenomenal. That's really, and this changes a lot. Okay? The entire biology changes, obviously the chemistry changes also. Okay, so I think it's important to recognize that minor structural differences actually make a lot of difference. All right, so we will do N-glycan and O-glycan structures a little bit later on. The oligosaccharides are really important for cell-cell interactions, for agglutination, hemagglutination, things of that sort that play a major role. And we will do this in a little more detail, so if I skip right now, I think it should be fine. Normally, we represent the N and O glycans by using symbols, and there is no real nomenclature that has been deduced. Various institutions have proposed symbols to represent these monosaccharides, and these symbols are heavily used in the book that I'm going to give out to all the trainees. Okay, so this is a book that Dr. Ajit Varki of University of San Diego has compiled. It's a beautiful book. I'm going to hand out to each of the trainees. We don't have all the copies yet. The secretary tried to get all, but we have received only 7. So my students will get it, and my place, and Kubi's students will receive it later on. The rest of the people will be able to take the copies. Go ahead.
Do you have like an e-book?
E-book.
Yeah.
Yeah. So this book is for free. It is available on the internet.
Okay.
So and yet I want you to have the hard copy. Okay, primarily because it's a good reference book and it describes a gamut of the processes in which carbohydrates play a major role. All the structures, biology, a lot of a lot of things about carbohydrates, and it's a good reference. And this particular book uses this nomenclature, for example, to describe that particular structure. So while you can write alpha, D, 1 to 4, galactose, N, P, 2AC, all of that, what it tries to do, it tries to convert it into this form, assuming that certain bonds, interglycosidic bonds, are either normally only of one type in nature or can be written on top of this in symbols such as 2, 3, and things of that sort. So in this particular case, for example, you would have fucose is denoted like that. The mannose is typically denoted, and then the colors also mean something. So, and you have to follow that convention, all right? So this is a standard way nowadays to write on top of mass spectra, for example. When you have a mass spectral pattern, a given peak has a defined structure, and you write this sort of a symbol. It makes it easier for people to understand. So it's important for you to, obviously, I don't remember all of this. And the number of symbols that are available are too few for the number of monosaccharides that are actually present in nature. They are just phenomenal. So we would run out of the number of symbols. So it's important to be able to use the alpha D 1, 4, the entire description in general. But it's also important for you all to familiarize with this, primarily because some authors tend to use this. So it's important that we know everything. Okay, let's stop here. Let's see if Kubi wants to take on from here.